Hey everyone, just picking up where we left off. We've talked a little bit about autoimmune disease in the past, but autoimmune disease is defined by the self being identified as foreign. So the immune system is meant to distinguish between self versus foreign, and autoimmune disease is when the self is identified as foreign. We have a few examples here. One example that some of you may be familiar with is celiac disease. It's an autoimmune attack against your intestinal wall, and it's primarily exacerbated by consuming gluten-rich food. Another example that a lot of you are also probably familiar with is type 1 diabetes. What's unique about this as an autoimmune disease is that the pancreatic beta cells that produce insulin are identified as foreign and undergo an autoimmune attack and are destroyed, and is how insulin is no longer produced among those suffering from type 1 diabetes. Another one is multiple sclerosis. It's a progressive disease of neurodegeneration where there's an autoimmune attack. There's a part of our central nervous system that is identified as foreign, attacked, and promotes the slow and steady neurodegeneration associated with multiple sclerosis. And finally, rheumatoid arthritis. It's a broadly systemic disease and can actually affect uh, many organ systems, primarily the joints, but it's able to affect multiple organ systems primarily because the autoantigen, the molecule that's self and identified as foreign, is collagen here. And as we've talked about previously, collagen is the most abundant protein in our body, and so um, it's going to lead to autoimmune symptoms that extend beyond the joints. One possible etiology or one possible cause of autoimmune disease is this idea of molecular mimicry. That's the idea that some antigen can initiate an immune response that resembles or mimics some other antigen. And so in the case of autoimmune disease, there's something that is self that mimics an antigen and produces this autoimmune response. And what ends up happening is that the antigen receptors or B cell receptors or T cell receptors and antibodies will cross-react, meaning they'll bind to something they're not supposed to bind to. In this case, what they're not supposed to bind to is a self protein. This self protein mimics some antigen and is now going to be dubbed an autoantigen. And so this autoantigen the idea of this autoantigen emerging might be because of molecular mimicry. It mimics some antigen that's actually pathogenic. And when this happens, now our immune system will recognize some part of ourself as foreign and then lead to some sort of immune response that leads to the inhibition of that protein or perhaps attacking and destroying the cells that express that protein. So coming all the way back to Guillain-Barré syndrome, um, most cases for that syndrome will begin about a week or two after there's an infection of the C. jejuni. Um, and it will typically begin with enteritis, which is inflammation of our small intestines. And the reason why we're talking about this is because this is an instance where mimicry is part of the cause. So when it comes to autoimmune disease like this, we want to consider what the antigen is, how is the antigen presented, is the immune response humoral or cell mediated, and what in fact is the autoantigen here. Um, One thing to consider here is uh, a lot of the things we've discussed about the immune system. We're talking about fundamental immunobiology, the fundamental building blocks of the immune system. And so we're talking about things that are by and large generally true. When we take things to um, another layer, when we take things to like another degree of complexity, typically what happens is that there can sometimes be exceptions to the rule. And so what we've talked about so far is that proteins that are presented are usually presented on MHC proteins, MHC class 1 and MHC class 2. There's a unique instance in immunobiology where there is another molecule, surface molecule that will present molecules. In this case, CD1 is actually able to present not proteins, but in this case, lipids. And so what ends up happening, the way this connects back to C. jejuni is that um, when or the way that it connects back to Guillain-Barré syndrome, forgive me, is that the C. jejuni bacterium is ingested and will cross the intestinal epithelium. There's going to be a B cell where their B cell receptor recognizes some epitope on the C. jejuni antigen. It will internalize it via receptor-mediated endocytosis. It will process it and display the lipid contents on CD1. Um, the lipid molecule that's presented on the surface will then be recognized by some T cell population. And in particular, it's what's presented on CD1 on this B cell is called a lipo oligosaccharide. Again, oligo means many, saccharide's a sugar. So this is a lipid molecule with sugar groups added onto it. Once the uh, T cell that bears the T cell receptor that recognizes this lipooligosaccharide, it will then trigger the T cell to activate the B cell. The B cell will then act on the T cell. And once the B cell is activated by cytokines secreted by the T cell, it'll trigger the B cell to differentiate into a plasma cell and start releasing antibodies that are against or recognize that lipooligosaccharide. Um, the autoantigen, this lipooligosaccharide that is recognized in the case of Guillain-Barré syndrome, is part of a family of lipids that have these sugar groups on them. We call them gangliosides. What's unique about these gangliosides is that there's a certain feature or epitope on gangliosides that are on found on C. jejuni that resemble a component of our Schwann cells that are part of our peripheral nervous system. And you can see that's boxed in here, and they're nearly identical. And so they're enriched on Schwann cells. Gangliosides are enriched on Schwann cells because of their ability to help promote and drive signaling in that cell population. And so these autoantibodies that recognize gangliosides will act on Schwann cells and inhibit their function. What's really key about inhibiting Schwann cell function here is um, that Schwann cells are the key cell population on our peripheral nervous system that secrete myelin and wrap them around our nerves that enables saltatory conduction across our nerves. And so by attacking Schwann cells, this will lead to the loss of myelin on our peripheral nerves, and that will inhibit or slow down nerve conductions. Um, and so that's the connection here between this bacterial infection that leads to this autoimmune attack that targets our peripheral nervous system that reduces neuron activity and the ability for conduction to happen down a peripheral nerve. As far as treatments for Guillain-Barré syndrome, as you can imagine, when we're talking about our peripheral nervous system, that affects a lot of key biological functions. Chief among them is the ability to breathe. And so typically patients with Guillain-Barré syndrome will need some sort of respiratory support. And so they'll be in these respiratory support, um, almost like an iron lung from a while back, but an opportunity to try and get support to breathe appropriately. Patients will also get what we call plasmapheresis. It'll be a treatment that helps to try and remove the plasma, the liquid component of our blood, remove the antibodies that recognize gangliosides and return the plasma back to the patient. Patients will also undergo immunosuppression therapy to try and avoid this autoimmune attack, plasmapheresis again. They'll also undergo immunoglobulin or antibody injections to try and neutralize and remove the C. jejuni bacterium from these patients. If the adaptive immunology lecture really caught your attention, there's a great cohort of people studying adaptive immunology here at UVA, and UVA has had a really strong history of having leaders in the field of T cell or B cell biology here. And here's a few individuals that are leaders in the field. And if this lecture really connected with you, I highly recommend you read some of their work and potentially reach out to them for research opportunities. Thanks, guys.